This is the second of 3 videos looking at the electron transport chain and oxidative phosphorylation. So if you haven't seen the first video, it might be worth checking that out. But I'm not going to tell you what to do, so if you want to just jump right into it, let's jump right into it. So what we're going to cover in this, in the previous video, we talked about what is an electron transport chain. In this video, we're just going to go through the reactions of the electron transport chain itself. And the idea here is that if you understand the mechanics of the transfers that are happening, you'll be better prepared to understand how it works and why it works the way that it does. So quick reminder, or I don't know if I've actually said it to make sure we're on the same page, the electron transport chain occurs between the mitochondrial matrix and the intermembrane space. So here I've got a picture of a mitochondria. Mitochondria have this double membrane structure. There is an outer membrane that's fairly permeable to the cell, and then there's an inner membrane that is very impermeable to things crossing that bilayer without transporters. And the electron transport chain is happening in this bilayer here, on this bilayer between on the inner membrane between the matrix and the intermembrane space. So the matrix is here, the IMS is here, and the electron transport chain is happening in that region there. What happens in the electron transport chain is electrons flow from electron carriers to molecular oxygen by going through a variety of complexes. And this is all happening embedded within that inner mitochondrial membrane. And the functional, or the process, I guess, description of what's going on is the electron transport chain passes electrons from one mobile carrier to the next. So we can put electrons on a mobile carrier like NADH. We're going to transfer them to a mobile carrier QH2 at complex 1. That QH2 then diffuses over to complex 3, which then transfers the electrons from QH3 onto cytochrome C. Cytochrome C then diffuses over to complex 4, and at complex 4, we take the electrons from cytochrome C and we use them to reduce molecular oxygen. This is then coupled to this proton transport process where we're taking protons, 4 protons at complex 1, 4 protons at complex 3, and 2 protons at complex 1. So we're pushing protons across the bilayer, being driven by this oxidative reaction. And so what I'd like to do in this video is, in a little bit more detail than I did in the last one, which was kind of a high-level survey, it's what are the mobile carriers, what are the reactions at each complex, and then if we summarize the whole thing, what are the overall reactions of the electron transport chain, or what is the overall reaction? So let's get into that. What are the mobile carriers? And there are 3, whoa, that is not the right button. It's still not the right button. You don't want to see me. There are basically 3 mobile carriers that we're going to worry about here. The first is NADH and NAD+, which we've seen previously as an electron currency. We're going to talk about this one, QH2. This is ubiquinone, ubiquinol. And finally, we're going to talk about cytochrome C, which can be in the iron 3 or iron 2 redox state. And I'm going to show these as chemicals in a second, but I'm also going to show these graphically throughout the rest of this video in this sort of schematized form. Okay, so NAD, NADH. So this is nicotinamide adenine dinucleotide. This is used as an electron currency all across the cell, where cells carefully maintain a high ratio of NAD+ to NADH. It has this oxidized form here where the nicotinamide ring has a charge on it, and then it has this reduced form where it picks up a hydride, a proton and 2 electrons, so it can now be in a reduced form. These molecules, this has a moderate affinity for electrons. So the standard state reduction potential for this is minus 0.315 volts, which is a moderate electron affinity. This will pick up electrons, but it will also fairly readily donate them to other reactions. So this is the first mobile carrier, and what it's going to do is basically connect things like the citric acid cycle into the electron transport chain. Another thing we might want to note about this is that it's very water soluble. If you look across it, there's a bunch of charges and there's a bunch of hydrogen bond donors and acceptors. So this will readily dissolve in water. So it floats around in the aqueous cytosol and matrix and intermembrane space of cells. Okay. This contrasts with ubiquinone, ubiquinol, which is Q and QH2. So the ubiquinone form is the oxidized form. The ubiquinol form is the reduced form. This is also known, just so in case you come across it, as coenzyme Q, also Q10. There's a variety of different names for it. I will consistently refer to this as ubiquinone, ubiquinol, or Q and QH2. This molecule can take an electron into its ring structure, much like NADH can. In this case, we have its oxidized form over here. You take 2 protons and two electrons. Oops, I have the wrong form. This should have an H. Sorry about that. Typo there. This has the H form in it, so this is 2 electrons and 2 protons, 2 electrons and 2 protons, but you go across this here, and now you've stored 2 electrons in that ring structure. It also has this long 10 chain here, which when you draw it is really, really, really long, right? So this is a long, greasy hydrophobic thing. And what this means is unlike NADH, which likes hanging out in the cytosol or in the aqueous phase of a cell, QH2 and Q are always in membrane. So they're actually a membrane-bound form electron carrier. And they're going to move through the bilayer. They're going to carry electrons across the bilayer. They're going to carry protons across the bilayer because they're large, hydrophobic, and uncharged. Another important thing to note about these Q is the Q and QH2 is it can actually exist in a semi-ubiquinone state as well. And so what that state is instead of taking multiple electrons at once, we have a single electron that is then shared across a variety of resonance forms in this structure. And it turns out that's going to be really important for its functions, because one of the things that Q does is it takes single electrons and passes them on to ions, which take single electrons as well. So Q ends up being kind of an adapter between things like NADH, which are kind of obligate 2 electron moving molecules, to iron, which are obligate 1 moving molecules. A Q can move 2 electrons or 1 electron. All right, that gets us to our last electron carrier, this cytochrome C. Cytochrome C is a protein that has a heme embedded in the middle. You may have seen hemes before with, say, hemoglobin. What this is the ring structure, a porphyrin ring that holds an iron in the middle. And it turns out that iron can flip between oxidation states. It can flip between an iron 3 and an iron 2 state. So when it's reduced, it has a less positive charge because it's got that extra electron, and that can be stably held on to by this iron. In fact, the energy to do this is much higher than for the previous things. Actually, I don't think I said on the last slide, but the standard state reduction potential for Q is higher than NADH. This iron is higher still than Q. So what that means is electrons in the electron transport chain are going to flow from NADH to QH2 to iron 2 to cytochrome C in the 2 form. Now, it might not be obvious from this picture here, but this actually skates along the membrane. So this protein actually gets a hydrophobic bit attached to the bottom, and that embeds itself in bilayers. So when we're thinking about this as a mobile carrier, what we can actually think about is our cytochrome Cs are moving along the top of the bilayer, skating along the surface, carrying their electrons to whatever destination they're going to. Okay, and lastly, this isn't really an electron acceptor terminal. This is not a electron carrier, mobile carrier. It's the terminal electron acceptor, but I wanted to put it on there to illustrate the last step of this. So what this is building towards, all this electron transport chain, is taking electrons from NADH and then passing them to QH2 and then passing them to cytochrome C, and then ultimately we're going to pass them onto oxygen into water. And this is the terminal, what's known as the terminal electron acceptor. Electrons aren't going to go any further. We fully oxidized the NADH or whatever comes in at this point. And just to continue to hammer this point home, this has an even higher standard state reduction potential. This is a higher electron affinity. So this is sucking electrons down the electron transport chain, ultimately sticking them here into water. Okay, so what are the mobile carriers? We just went over them. They're NADH, QH2, and this iron 3 cytochrome C, or iron 2 cytochrome C, sorry. Next, we're going to ask, what are the reactions that occur at each of the complexes in the electron transport chain? And the first thing we want to think about is QH2. I said that was our second electron, or our second mobile electron carrier. There are actually 2 sources of QH2 in the electron transport chain, 2 independent sources. So what we're going to do is look at source 1 and then source 2. And what we're going to be asking is, how do you go from this, the Q form, to this, the QH2 form? So complex 1 first. So what happens in complex 1? So complex 1, what happens is you're going to transfer 2 electrons from an NADH to a QH2. This happens at the matrix side of the bilayer. So here's the matrix, here's the IMS up here. And it's a fairly straightforward reaction. We're going to take 2 protons from the matrix and 2 electrons from our NADH. we're going to stick them together and use those to go from Q to QH2. And then we're going to release our oxidized NAD plus molecule here. This is also associated, the 2 electrons came from the NADH, the 2 protons came from the matrix, and this is also associated with pumping 4 protons from the matrix into the IMS. And I'm not going to go into the mechanistic details of this. It's actually a really cool mechanism that's only just now being worked out. Look for a future video on that. But for now, we're just going to say the complex, through the magic of allostery, actually pumps 4 protons across the bilayer in a way that's coupled with this oxidation reaction. Okay, there's complex 1. What's the net reaction for this? Well, we're going to take 4 protons from the matrix. We're going to take 2 other protons in the matrix, and then we are going to run a reaction where we move 4 protons into the intermembrane space, so across the bilayer. We're going to take 2 of the protons from the matrix, 2 of the electrons from NADH, and we're going to ultimately generate a QH2 using everything from the matrix, not the IMS. Okay. Complex 2. I said there are 2 sources of QH2. Complex 2 is an alternate source of QH2. And what it does is you take 2 electrons from something from the molecule succinate, and you take 2 protons from the matrix, and you use those to generate a QH2. So 2 electrons from succinate, 2 protons from the matrix. No protons are pumped from the matrix to the IMS. So there's no proton pump like there is at complex 1. And if you're thinking, man, I think I've seen succinate before, it's because you have. This is actually part of the citric acid cycle. Down here on the bottom left, we take a succinate and we oxidize it to a fumarate. And I said, if you watched my video on that, I said, oh yeah, we'll use that to take a Q to make a QH2. You are reducing this mobile electron carrier, ubiquinone, to ubiquinol using succinate as the substrate. So this is part of the citric acid cycle, which is occurring inside of the matrix of mitochondria. Okay, so net reaction of this, you're going to take 2 electrons from succinate, 2 protons from the matrix, and you're going to generate fumarate and your reduced QH2. Okay, so I said there's 2 sources of QH2. We've generated these mobile electron carriers QH2. What do we do next? What do we do with those in future complexes? So it turns out we go to complex 3, and this is one of the more complicated sets of reactions. In fact, I'm showing a simplified form of it. What we're going to show is something called the Q cycle. And the overall idea of the Q cycle is to take electrons from Q and put them onto cytochrome C. And the reason it's complicated is because, remember, Q has 2 electrons, or QH2 has 2 electrons, and we're going onto iron, which can only take 1 electron. So there's a complicated machine that achieves this. And again, this is something I'm not going to go into too much mechanistic detail on, but look for a future video on that because it's a really cool mechanism. Okay, but here's the simple form. I think I actually have to disappear because there's going to be a lot of stuff on these slides. That's the wrong button. There we go, right button. Okay, so what happens here? So remember, ubiquinol, or this QH2, is in the bilayer, right? It's extremely hydrophobic, so it's floating through the bilayer. And that cytochrome C skates along the bilayer. The cytochrome C is actually on the IMS side. And so what happens is QH2 comes along and donates one of its electrons up here onto an iron, forming iron 2 in a cytochrome C. It also takes 1 electron and goes back into complex 3. So it actually takes those 2 electrons off of ubiquinol, making ubiquinone, going from QH2 to Q. Both electrons come off. 1 electron goes onto cytochrome C, the other goes back into the complex. And finally, the 2 protons that we picked up when we reduced Q to QH2 now get dumped out on the IMS side. So basically, remember, we loaded those over here on the matrix side. We dumped them on the IMS side. So we're doing a net transport of protons from the IMS, or from the matrix to the IMS, through Q as the mediator. So QH2 is not only acting as a mobile carrier for electrons, it's also acting as a mobile carrier for protons. Okay, so now the question is, what do we do with that electron that went back into the complex? So that electron, it turns out, goes into another Q that is in its semi-ubiquinone form. So it has a single electron loaded into it. And that semi-ubiquinone form is actually generated by another process. So there's like, this happens twice in a row. So you can think about one electron leaves a QH2 and goes into a Q to make a single electron. And then we see this happening again. So we're basically seeing the second event, the second time a QH2 is reduced, it loads that electron into a semi-ubiquinone, creating another QH2. But now the electrons, one came from the first one, another electron was already there in the complex. And it's going to grab onto its protons from the matrix. So now you've made the QH2 using this electron and these protons in the matrix. What happens next is exactly like what we just saw happening there. We're going to take, we load that electron onto the existing radical, and we move 2 protons from the matrix onto QH2. The last step looks a heck of a lot like this one. So if this is step A, this is step B, right? And in step B, we're taking our QH2 and we are pulling the electrons out. One of the electrons goes onto our cytochrome C. One of the electrons goes down back into the complex where it's going to continue in the Q cycle. And finally, the 2 protons that we picked up down here on the matrix side are now dumped onto the IMS side. So we picked up an electron from QH2 and moved its iron. We moved an electron back into the complex to continue the Q cycle. And we took 2 protons from QH2 and dumped it into the IMS. What that means is we can actually write out a net reaction here that looks like the following. So we have a QH2 that comes in with 2 protons and 2 cytochrome Cs, which are in their oxidized form. And we have 2 protons that come from the from the matrix. When you do this whole complicated Q cycle looping thing, what you end up with is an oxidized Q, 2 reduced irons in cytochrome C, and you've moved 4 protons across the bilayer. So this whole complicated Q cycle thing in net moves 4 protons and loads 2 electrons onto cytochrome C. Okay, complex 3. Last thing we got to ask is, well, what happens to all that reduced cytochrome C that we generate at complex 3? Goes into complex 4. So this is hopefully making some logical sense. Complexes 1 and 2 generate QH2. Complex 3 generates cytochrome C. Complex 4 takes that cytochrome C and sticks the electrons onto oxygen. Now, it turns out, if you sort of think about the stoichiometry here, you're going to bring in 1 molecular oxygen. And if you think about the redox chemistry, what you're going to do to join this to 4 protons, you're going to actually need 4 new electrons to do so. So the way complex 4 works is it takes 1, 2, 3, 4 cytochrome Cs that were reduced by complex 3, takes those 4 electrons and sticks those 4 electrons onto oxygen and 4 protons, generating 2 new water molecules. So 4 electrons go from iron 2 onto 2 waters, and this then leaves 4 oxidized cytochrome Cs. The other part of this is we are going to move, by the magic of allostery, 4 protons from the matrix into the IMS as well. So if we think about what's the overall net reaction for complex 4, what we're doing is for 2 electrons now, we're taking 2 irons, 2 cytochrome Cs with electrons. We're taking half of an oxygen and 2 protons, and we're grabbing 2 protons from the bilayer, from the matrix. We're moving those protons into the IMS. We're oxidizing our cytochrome Cs, and we're generating a water. All right. Almost done here. So we ask, what are the mobile carriers? What are the reactions of each complex? Finally, what are the overall reactions if we sum up what happened there? And it turns out there's actually 2 reactions depending on where our QH2 came from. So first, if we come from NADH, we've got a complex 1, complex 3, and complex 4 evolved. And if we look at that overall reaction, we're going to take all the stuff I'm showing here on the left, and we're going to generate the things on the right. So we're going to take our NADH, we're going to generate an NAD, we're going to take an oxygen and some protons, we're going to generate some water, and we're going to move 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 protons across the bilayer. So NADH can drive 10 protons across the bilayer in the process of being oxidized. Succinate comes in, remember, at complex 2, and when it comes in, it takes the following. It takes 6 protons from the matrix, as well as 2 other protons. It then generates a water and a fumarate, and in the process moves 1, 2, 3, 4, 5, 6. It moves 6 protons. So it moves fewer protons than an NADH does. And it turns out you can actually rationalize this in terms of their standard state reduction potentials. NAD holds onto its electrons much less tightly than succinate does. So it's a bigger jump in redox to go from an NADH to an oxygen than it is to go from a succinate to an oxygen. So overall then, it becomes, you get more energetic bang for your buck going from NADH than you do from going from succinate. Okay, so what have we said thus far? We've said electrons flow from NADH and succinate to oxygen, pumping protons along the way. What we've done in this video is gone through and surveyed each of those individual reactions. In the next video, we are finally going to zoom out, or we're going to ask how in the world this actually leads to ATP synthesis at the ATP synthase complex. So hope you can join me for that. Thank you for taking the time now, and I will talk to you later.